All right. So the next talk is by Krishna Garikapati.
Thank you very much, Anuj. Um, just like Joseph, I also feel like an outsider in here, but not in a bad way at all. I'll explain what I mean. So anyhow, you see the topic of my talk. It's a mouthful, so I won't try to say repeat it. You also see a number of people have been associated with this work. Of this lot, Sid Srivastava and I recently moved from University of Michigan for many years on my part to USC. Uh, the rest of them, let me tell you, let me give you a little bit that'll probably place this talk in context. Uh, of the authors, including myself, many of us are what I would describe as computational scientists, and this means we model physics, we model phenomena, the physics of phenomena, using a combination of applied mathematics, numerical methods, computing, and now increasingly machine learning. All the rest of the people are biologists, right? So that's where we're coming together. Okay. The other bit about this talk is that it's fairly specific. It's one particular thread of work that we've been doing for a few years now. The way I wrote this talk, I do get into the mathematics a little bit, and I say that recognizing that 3 o'clock or 3:15 is about when caffeine becomes an important input. But maybe I can keep that at a higher level, and the process may perhaps spark some discussion here. Okay, so what are we trying to do here? We start out with these wound healing experiments in cancer cell migration. If you look at the schematic on the right, this tells you what's going on. This is called a scratch assay. You take a layer of cells, you literally scratch it, and that's shown there, and then you sit back and watch as this layer of cells then heals this wound, right? The thing that looks like, I don't know, Pac-Man or something like that on the left is actually a true a video made from a scratch assay. What you see as pixels are actually wells in which the cells have a sufficiently high density that you're actually seeing a reasonable density of cells in there. And what you're seeing is that they're actually closing this wound. What we're interested in doing here is trying to model this process from the standpoint of physics. And in order to do that, if you look at the last bullet point here, we do this by trying to understand the physics through mathematical operators, right? So this is what gets us into equations. It turns out for this process, we can model it through what we call mean field dynamics. The equation up here is an equation that's referred to as a Fokker-Planck equation with a proliferation term at it, right? So it also has a much longer name there, but that may or may not be of interest. Okay, so what are we trying to do here? C here represents the cell density, which means really what is the number of cells over a certain area. And then there are a number of other quantities, right? The Fokker-Planck equation describes the evolution of populations. It also describes the evolution of probability densities driven by a potential psi. D here, and these are all terms that appear in the equation, right? D here is a diffusivity. The potential is really understood as an external driving force that is maybe giving a directed motion to this population. The diffusivity comes from the idea that cells, like many other populations, also have a random walk aspect to them. And finally, R is from the standpoint of the equation, it's a reaction term, but it really models birth and death processes, right? So the cells are dividing and dying and so forth. Okay. I'll also mention that one can either write this mean field equation directly by observing these densities, or one can look at single cell dynamics, come up with a stochastic differential equation, and then systematically coarse grain that. Okay, so it doesn't matter how we do it. Okay. What's of interest to us here, to my group, is this last bullet point, where not only do we want to model this forward problem, but we also want to use it to infer parameters and mechanisms that are actually driving this physical process, right? So if you write an equation, you compare it with data, and you can infer things about what mechanisms are actually active in that equation. It tells you something about the process, right? So that's what we're doing. The way we go about this is essentially we take this equation, we write it out here in a different form. It's called the weak form, which is widely used which is used in finite element methods and other kinds of methods. Essentially, write the equation in a certain form, and you say that now the parameters that matter here, d star and the theta1 and theta1 and theta2 are vectors which go into determining this potential and this reaction term and so on. Anyway, you say that the optimal parameters are obviously obtained as argmins of this equation, right? And you can think about this as essentially the equation here represents a loss, trying to minimize that loss, and you're trying to find the optimal set of parameters. If you know these parameters, you know something about the mechanisms, you know something about the physics that's going. Okay, that's what we're trying to do. All right. So we've done this for exactly this wound healing problem that you've seen. We work with the Fokker-Planck equation with the reaction term. A simplified form of the Fokker-Planck equation is what is called the advection diffusion reaction equation. It's written here in this form. I've highlighted all the different types of mechanisms that are acting here. All right, let's look at some of these images. I'm not sure you can read all of that, but I'll tell you what's going on here. What we're showing here, plotting on the y-axis, is the density of cells along the y direction, and the horizontal axis is time. If you look at what you have here, you will see that you initially start out with fewer cells in the middle because that scratch is there, that wound exists. And then as time marches on, that wound gets filled, right? So you get these contours. Turns out if you follow one of these contours, you will get an equation, which I don't think you can quite see that. It turned out to be too small here, but it's essentially y to the power of y goes as time to the power of 0.4. Now, why 0.4? It turns out that if that 0.4 were 0.5, this would be a diffusion process, right? So now we're asking, well, is this a diffusion dominated or what? So we do our inference method, and after doing our inference method, we find an equation. So we parameterize this equation through inference, and then run the forward problem that we've found, right? We've inferred an equation, we run the forward problem, and we get a fairly good match with the data. This shows where the bursts of errors are. The errors of the order of a few percent. Essentially, I won't get into details here, but essentially what we could show is that diffusion does indeed dominate this process. The little bit of directed motion exists, but it's pretty low. Turned out that the reaction term, that this birth and death process, was also pretty small. Right, so one can infer this sort of thing, and we have an archive paper out there, papers under review. Right, so this is how we go about writing an equation for a process and then trying to infer what's going on in it. Right, and we use this. Okay. So this is now an image of a different type of experiment done with these same cells. This is called a chemotaxis assay. What you're seeing here are these individual cells. The two videos are the same. And on the left, you're seeing only the cell nuclei. And on the right, you're seeing the cells also around them. This experiment is done under a gradient of a chemoattractant, right? And that gradient is pointing leftward, which is what, and it turns out the potential is sort of decreasing to the left. But anyway, the cells are mostly moving leftward. But then if you look at it, you will see that there are a few cells which are not quite moving the same direction, like this one here, maybe that one too, not quite moving to the left. So what this shows us is that the cells have a certain heterogeneity to them, and they're really behaving as single agents, right, in here. Whereas the previous example I showed you was where the cells had a high enough density that we could really treat it as a sort of mean field problem. I will say that there's a great deal of interest in studying heterogeneity, and we're doing that in related work. I will also say that in much earlier work, we've looked at sort of single cell mechanics that makes these cells move in this manner. Anyway, so how do we treat this problem? This idea of cells behaving as agents now takes us to a slightly different approach here. And over the next few slides, I'm going to take this approach of Markov process, the Markov decision processes, right? This may be familiar to many people here, but I'll just introduce this. The idea is that you take each agent. Now, this is a cell. Each cell is an agent. It lives in a certain state action space, right? So state says that, well, you are in a particular state, and the action space says that, well, if you are in a certain state and you're going to make a move, what action do you take, right? So that's drawn from an action space. An important idea here is a transition probability density function, which says that if you are in a certain state in an action, what is the probability that you will be in another state at the next move? all of this is understood to be driven, or this is how the theory works, that this is all driven under a certain reward function, that the agents get a reward for occupying a certain state and action at a given instance. There's also another quantity in here, also probability density function. It's called a policy. And what the policy says is that if you are in a given state, what is the probability of you taking a certain action to get to another state? And it turns out that one can get an optimal policy by calculating an argmax over the reward, but counting not just for the current reward, not just the reward you get right now, but what is the expectation of you gathering reward over time? Right? So this is all done in this manner. Anyway, another important quantity in here, another important idea in here is the so-called Bellman equation. Are you kidding me? Okay, just 5 minutes. All right, which relates the reward to a different idea of value function. Okay, so I'm going to accelerate here to say that we also need to we want to approach this whole problem by looking at this inverse reinforcement learning. Inverse reinforcement learning takes certain trajectories and then comes up with a policy as well as a reward by going through a transition. All right. The challenge here is that the transition function is often difficult to specify here. It's common to obtain it empirically, but of course, when you obtain it empirically, it's just poor at extrapolation. Okay. The approach we take, and I realize I'm running out of time here, so let me just say that the approach we take is to take a step back and go back to a sort of Markov policy, to a Markov process, which allows us to club the state and action into a common space. The reason we do that is then we can, Markov processes are known to generate certain classes of equations, of which the Fokker-Planck equation is a member. That then allows us to describe this, go back to writing this process, this agent-based process through the PD, the same PD that we're aware that I was describing earlier. If you can then go back to that PD, the Fokker-Planck equation also generates a transition probability function, and that provides one of the important aspects that we need here, one of the important quantities we need here. There's a central conjecture to our approach which says that there is an isomorphism between the Fokker-Planck equation and Markov decision processes. And there are reasons why we make this conjecture. I won't get into it. There are lots of similarities between these processes. It also gives us a series of steps by which we can also come up with this policy for this Markov process, driven by the Fokker-Planck equation. Finally, we can put all these pieces together to also infer the reward from this process, right? And there's a theorem which, under fairly mild conditions, tells us that if we have a state value function, we can connect it to the reward. Anyway, okay. So we put all of this together. The important aspect is that, well, if I can. I need to be able to identify a Fokker-Planck equation here. If I can identify a Fokker-Planck equation here, do inference, that gives me a transition function, which then from which all of this flows. What does this all give me? It gives me a way to do inverse reinforcement learning by infusing physics into this process. The physics comes from the Fokker-Planck equation. All right. Just a summary of the algorithm. Okay, so let me tell you where we apply it, right? So we take this cancer cell dynamics, that last video I showed you. This is a readout of certain markers which our biologist colleagues obtain. And each streak in here represents a single cell. On the horizontal axis, you have time. Okay, so you have a number of cells, thousands of cells maybe, going up vertically, and in each of these columns represents that cell reporting a certain quantity. You're reporting a certain quantity from that cell over time. AKT and ERK are what are called kinase translocation reporters. They are kinases which have an important role. These are enzymes which have an important role in cell signaling and migration and so on. What you will see is that if you look at one of these kinases, right, ERK, it seems to be highly correlated with the speed here. You see the similar kind of colors and so forth. AKT, which is another kinase, seems to be less related. So we're trying to ask, what is the relation between these? For our state and action space, we take the states to consist of the velocities that the cells are following. The action space is which of these kinase translocation reporters are you expressing? Okay, so we run our inverse reinforcement learning process, and here's our result. Never mind what's happening on the right. Let's look at what's happening on the left. The horizontal axes represent the states. This is the velocity in the x direction. That's the velocity in the y direction. I'm plotting up the reward. Let's focus on this region where the reward is high. Within each of these boxes, there's a smaller box in here which reports the values of the actions taken, right? So what is the ERK and AKT expression by these cells? I won't get into details of how to interpret this, but then it turns out that we infer from our inverse reinforcement learning that a higher reward is obtained by the cells for moving leftward, so that the velocity Vx is negative, so it's moving leftward, and slightly upward, Vy is slightly higher, right, positive. So it comes into this region, and furthermore, for expressing low levels of one of these reporters, AKT, and high values of another reporter, that's ERK. How does this correspond with experiment? This is a series of experiments run by our biologist colleagues. Each of these spaghetti-like lines that you're seeing shows a cell trajectory, right? And what you're seeing is that on the average, cells are moving leftward. This is the control experiment. There are a number of interventions they do. This one shows the suppression of ERK. And when ERK is suppressed, we see that these trajectories are very much decreased. This shows the suppression of AKT, and the trajectories are slightly higher with AKT. Last two slides. How does this relate to what we're doing? What we found was that that we need higher values of ERK and low values of AKT from our inverse reinforcement learning. What these experiments show is that when ERK is suppressed, we get much lower trajectories, right? Much lower velocity. Sort of a double negative here. So they suppressed ERK, found lower velocities. We found higher ERK is needed for higher velocities. So that corresponds with it. In this case, they suppress AKT and found slightly larger velocities. This plot here just shows quantifies the fact that suppressing AKT did indeed lead to higher velocities. I won't get into this, but anyway. So that's it. What I've described here is an approach by which, first of all, we can infer PDEs, which describe cell dynamics, and this talks about the Fokker-Planck equation. This led us to the Fokker-Planck equation. A number of ways in which we can arrive at this Fokker-Planck equation. It provides a probabilistic framework for inverse reinforcement learning, and the idea is that if the reason we go to inverse reinforcement learning is to try and understand the reward function for these cells, and so try to understand, can we infer this using the Fokker-Planck equation here infuses a certain amount of physics into these inverse reinforcement learning algorithms. There's this idea of the structural isomorphism between Fokker-Planck and Markov decision processes. Even from the standpoint of the algorithmic questions here, taking this sort of approach infuses greater computational efficiency. It turns out that IRL algorithms have a number of iterations in them, whereas what we have is a one-shot process. There are certain limitations. What I've presented here depends upon the populations actually following these certain physics. Obviously, physics has to be reflected. Not all processes have this. There's a single agent assumption and so forth. And there are further steps where we can expand into other types of physics described by transport theory. And of course, there's a lot of uncertainty quantification to be done. The top line are the people who actually did the work, the postdocs and students, and these are my collaborators. Thanks.
Let's thank Krishna for the wonderful talk. And we have time for questions.
No, nice talk. I think maybe that's what I'm looking for. What are the physics more into the process?
Yeah.
Now, you got some experimental data which are controlled experiments.
Right.
If you have a lot of data, what is that going to? Is that going to help you if you have a lot of data?
Yes. With these kinds of biological experiments, for the second set of these data, are actually they're of the order of about hundreds of cells, but this is over 24 hours. So yes, absolutely. With more data, our methods just get more robust. For instance, if you have experiments in which you're looking at cells in this particular case, and you have a sufficient density of cells, then you can directly go to these mean field assumptions, right? You can really say that, yes, I can describe this as a real cell density. In other cases where we have much sparser experiments, where there are fewer cells itself, one has to do other things. One has to sort of take a different approach where you really treat the dynamics of single cells and then take systematic coarse graining approaches where we go from stochastic differential equations to PDEs before we can use those PDEs in informing these reinforcement learning algorithms.
That's a wonderful talk. Thank you. So I'm kind of curious. You're looking at the expression of ERK and AKT. So these are signaling proteins,
Exactly,
kinases.
yeah.
But the movement is the motor proteins, right? The kinesins and
Yeah.
So,
Absolutely.
but there are other ODE cascades between ERK
Absolutely,
and the motor proteins, which will have a feedback control on ERK itself. So there's a much more complicated mechanism sitting behind this.
Absolutely, very true. Right. So what we're seeing here, so this is actually a much higher dimensional space than even we're exploring. So we're essentially looking at a probability density function that's evolving in some high dimensional space. Right here, I'm showing you 4, but actually there are very many more. So we're not hanging our hat on the absolute. We're not claiming with any great confidence yet that the causality that we're seeing here is really solid. One would have to have that entire cascade in place. And of course, something that came up often during the earlier talks is there's a lot of correlation in there also, right? So ultimately, we would want to get that whole, look at the much higher dimensional space and figure out, figure out causality, correlations, and maybe project from there into a parsimonious lower dimensional space to describe this process.
Let's thank Krishna again for the wonderful talk.